This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 284, recorded on March 16th, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Is it Chamber of Commerce weather there yet, or still cold?
It's a little cool today, but we're supposed to have 80 degrees tomorrow, and the sun is out, the azaleas are blooming, the dogwoods are blooming. It's really quite pretty.
I was just in Atlanta, and I thought it would be warm, because you're always saying the South is warm. It was freezing.
We're on the coast. Atlanta might as well be in Tennessee, if you think about it.
It was below zero. Also joining us from St. Louis, Missouri, Petra Levin.
It's good to be here. It's good to be here. I think it's sort of springish today, but it's gone back to winter. I think we're supposed to have 21 degrees Fahrenheit on Sunday. I know. The magnolia trees are blooming, but they're not very smart. They always go when it turns warm, and then they get hit. So
yeah,
I'm trying to appreciate them right now.
Yeah, Missouri's north of Georgia. Yeah. I mean, Georgia's right by Florida. I thought it would be warm, but... Man.
But Georgia's a very large state. Before Texas, Georgia was the largest state in the Union.
All right. This is what we have for you today. We have pigs and squid.
Pigs and squid.
It's not a restaurant. It's microbiology, and it's two fun papers. So Michael is going to start us off with pigs.
Pigs. This paper is entitled Bacterial Genome Sequencing Tracks the Housefly-Associated Dispersal of Fluoroquinolone and Cephalosporin-Resistant Escherichia coli from a pig farm that happens to be in Germany. And it appeared in Applied Microbiology on 6 February 2023, and it's an open source document, so you can follow along if you'd like. And the manuscript was authored by Behrens, Dolsdorf, Amann, Kolte, Borger, Werner, Juncker, and Frentrup. And they're at a number of institutes over in Germany, and I will butcher the names, so I won't insult them by trying to pronounce them. But since it's an open source document, you can take a look at where they're from. It will not likely surprise many of our TWiM regulars that the regular use of antimicrobials in livestock production selects for antimicrobial resistance. We have previously discussed this topic on other TWiMs, specifically the volume of antibiotics used in the production of our animal proteins, whether they be poultry, pork, lamb, or beef. It's measured, the concentration of antibiotics used in the production of that livestock is measured in the metric tons per year, and of course is a major contributor to antimicrobial resistance. In fact, I've dropped into your show notes today a data note that appeared in Nature on 3 February as well, and it's offered by Sarah Reardon, that offers that the use of antibiotics in animal farming is expected to grow by 8% between 2020 and 2030, despite ongoing efforts to curtail their use. Now, while 8% may seem modest, given our desire for animal protein in our diets, especially in emerging nations, one needs to consider the tonnage, which is estimated by the end of the decade, we will be using 107,500 tons to produce livestock per year, principally to reduce the farm to table time compared to just under 100,000 tons in 2020. But for those of us who don't think in tons, normally tons is an abstract, that's 220 million pounds of antibiotics. The data that are in the data note really are pretty interesting. They tell which country is the number one consumer of antibiotics for veterinary micro. And surprisingly, the United States is not number one, but rather we're number three. We're trying hard though. We're outpaced by number two, which is Brazil, and number one being China. I also dropped into the show notes. They offer a great figure for this, and so you can check out the Nature data note, but unfortunately it's behind a paywall. And there's another PLOS paper that is in, let's see, what is it called? It's in PLOS Global Public Health that actually describes the global trends in antimicrobial use in food-producing animals. And that's where they got the data for the data note. And it's a pretty interesting read. Now, don't misinterpret. I'm not advocating not to use antibiotics to treat infections in livestock. I'm not heartless, but rather I'm suggesting, as others are, that we don't need to use them to reduce the farm to table times. And furthermore, we really need to rethink the use of antibiotics to limit disease amongst animals in crowded and unsanitary conditions. But then that's a discussion for another TWiM.
So one of the interesting things about the antibiotics in animals was it wasn't only to treat disease. They found that actually giving them antibiotics early on, they got bigger and they grew faster, which is still, I think, a little mysterious. So it wasn't even like we're trying to keep them from getting sick.
It's farm to table. It's a farm to table.
If they fed them an antibiotic which was useless because there's a lot of resistance, would it still have the same effect on growth?
It does.
So
because
it makes inhibiting microbes, right?
Well, it does inhibit microbes, and it's all about nutrient availability because you're starving the microbes because the antibiotics will send them into a V, B, and C state. And then the nutrition that the animal is consuming, rather than feeding the microbes, will feed the animal. And that's what the magic of farm to table is all about. So today's snippet, as the title offers, that genome sequencing tracking the housefly, specifically looking at the associated dispersal of a fluoroquinolone antibiotic and a cephalosporin resistant E. coli from a pig farm, is pretty much the intent of this paper, namely how our current practice of using antibiotics to promote animals to develop our protein does indeed impact human health from the perspective of the role of the environment for the persistence and transmission of antimicrobial resistant bacteria. And this is just not confined to the U.S. This is across our globe. Among the potential linkages between animal health and human health and the environment, this has been previously addressed by the One Health concept. If you're interested in that, there's a lot written out there about One Health and how zoonosis is actually impacting on human health. After all, we just lived through COVID, which is really a One Health concept as the SARS-CoV-2 jumped from bats into people. And so you may want to think about that as we're laying out the story. Among the pathways considered for antimicrobial resistance from E. coli from livestock reservoirs to humans are simply the consumption of contaminated meat products. And many in the United States have appreciated that we have tried to begin to sell antibiotic-free beef, antibiotic-free pork, as well as chicken. And in fact, many of the fast food chains are priding themselves at selling antibiotic-free poultry in your chicken McNuggets. But there are also other ways to get these antibiotic resistance traits into the human population. And those are environmental routes, such as aerosols. Like when you flush your toilet, that's going to generate aerosols. And if you happen to have antibiotic resistant microbes in your stool, they could be flying around your house, or via vectors like insects, specifically houseflies. Now, there is some good news in that the airborne spread of E. coli is limited by the pathogen's low desiccation tolerance. But the problem with the common housefly, Musca domestica, has been identified as this vector of antimicrobial resistance in E. coli and other diverse pathogens. So in our paper today, we're going to learn about the movement of antibiotic resistance from a pig farm in Germany to its urban surroundings around Brandenburg, Germany. Cutting to the bottom line up front, the authors detected abundant cephalosporin and fluoroquinolone resistant E. coli in pig feces, sedimented dust, the dust that settles in our house, as well as our barns and livestock pens, as well as houseflies. They effectively captured flies, crushed them, extracted the DNA, and found the bacteria with these antibiotic resistance traits. This is pretty wild. And principally, the results of the paper showed from the genomic sequencing of E. coli isolates, they revealed a large phylogenetic diversity and plasmid-borne extended spectrum beta-lactamase, or ESBL. And if you've been reading anything that's been coming out of the CDC of late, you know ESBL is one of the big buzzwords, as well as the CTX-M1, which is the cefotaximine resistant strain. And again, these are beta-lactamase antimicrobials, which principally are interfering with peptidoglycan synthesis. Now, these phylogenetically close relationship, as measured by simply looking for changes in base pairs and comparing them, suggested frequent transmission of these antimicrobial resistant coli between pigs, as well as from different herds. Different herds means the little piglets come into these finishing facilities where they grow them up to a size to be sent to our table. And they have different breeders breeding them. And many of the piglets are given antimicrobials very early on in life to prevent diarrheal diseases, because they can literally wipe out if you will, a herd of pigs that you're hoping to fatten to take to market. Now, the result that really slapped me upside my head, especially when the authors reinforced that they were recovering from the farmyard resistant E. coli, but they were actually recovering resistant E. coli from flies that they collected 2 kilometers away from the farm. In the city. You heard correctly. 2 kilometers from the farm, it was detected in the flies.
I didn't know flies could go that far.
They can.
2 kilometers. Wow.
That's a pretty good distance. Most over 50s can't run that far. Or if they do, they'll be dead.
The dust also was kilometers away?
No, the dust was in the barns.
Okay.
The dust was in the barns.
So this is true. This is like what my mother told me, that flies are dirty, right?
Yes,
so
your mother was right, Petra.
Sure, they're dirty, yeah. They sit on feces, right? Flies are always doing that.
They eat them. They're coprophagic. They eat stool.
Yeah.
This morning, I gave a lecture to the med students about the microbiome. And I was presenting that paper that we had on TWiM a few years ago about autism in the mice. Mm. And I told them it was really easy to demonstrate Koch's postulates because you feed stool from mouse population 1 to mouse population 2, and the mice eat the stool. And the med students were grossed out by that. Well, anyway, back to the story. Why flies? Well, the common housefly is, again, coprophagic. That is, they feed on feces and manure, which, if you've ever been to a pig farm, there is stool everywhere. And that same buzzing fly around barns, and there's loads of flies in barns, is the same fly that sneaks in our house. So Petra's mother was right. Maybe that's the title of the show for today. Petra's mother was right.
She would enjoy.
Yes. And flies are synanthrope. I can never say that word. They live with us. And remarkably, the data from this paper offers that from the genome sequences they evaluated, they were identical or closely related to the pig feces that they isolated on the farm, indicating that the fly-associated transport of these very bizarre, diverse ESBL, and much of the data goes in MLST sequence typing, or they're using other methods to effectively figure out who begat who. It reads like the book of Genesis. You figure where the lineage came from. And the principal takeaway from all of this is that they observed the proximity of contaminated flies to human household poses a risk of transmission of antimicrobial resistant enteric pathogens from livestock to man. And the authors analyzed the genome sequences they recover, and remarkably, they were able to track them to the origin of the piglets. The other remarkable data element from this paper is that of the 3 antibiotics they screened, their resistance profile all increased as a function of time on the farm. It's really no different than what we see in a hospital setting. You're more likely to acquire an antibiotic-resistant strain the longer you stay in a hospital. And the 3 antibiotics that they looked for all have different microbial targets. The fluoroquinolones target gyrase and topoisomerase, and they were looking for point mutations. The cefotaximine targets peptidoglycan. It's a beta-lactam antibiotic. And the colistin goes after the LPS and the outer membrane of gram-negatives, as well as its cytoplasmic membrane, leading to the disruption of the cell envelope. And they had 12 herds that they studied at their fattening farm. And the data are pretty convincing. As you go out in time, the antibiotic resistance increases, and it's graphed on a logarithmic scale. And this increase is seen irrespective of where the pigs came from. So it's not one bad breeding farm that is sending them all the bad actors. And frequently, pigs from different herds can actually leave their stool, and then the dust and the flies serve as dissemination for more fecal pathogens. And the barn dust may actually carry fecal particles, including bacteria, which then may get ingested or inhaled by the pig and subsequently become established in the pig's gut or the human host that's caring for the pigs and mucking out the pens. Now, I want you to think about this the next time you take your kids to a petting zoo or even the zoo, as you're eating that cotton candy or snacking on the Cracker Jacks as you wander through the enclosures. I mean, this blew my mind. And I was reading this on the airplane coming across the country yesterday. And the poor woman sitting next to me, she said, "What are you reading?" She saw the title, and so I had to explain to her what TWiM was all about. She found it absolutely fascinating.
Did she put on her mask when you were talking to her about
No, she didn't put on her mask. No one on planes is wearing masks. Now, these stool-eating flies are commonly contaminated with gut bacteria, since both the adult fly and the larval flies, again, feed on animal feces. So, it's really the issue of the fly and the flying distances. And flies can go several kilometers per day. And these have been individually documented by effectively painting dye on the fly, capture the fly, paint some dye on it, and then release it. And they look for the dye that's coming back. And it's really quite fascinating, the care and effort that went through. All of their genetic analysis is really elegant, and it doesn't lend itself to radio or a podcast per se. You really need to look at the figures. And they talk about the detection of E. coli and Salmonella, and they offer that they have indistinguishable pulsal types, which is a DNA macro restriction pattern that's resolved by pulse field gel electrophoresis, which is sort of falling out of favor given the drop in price of DNA sequencing. So it's really changing. So I know much has been made about the zoonotic transmission of viruses to humans, but what they offer is perhaps we should be screening flies. And I don't know whether or not the flies could be a much better sentinel. They're certainly much easier to capture than bats. So maybe that's our best hope at trying to understand the movement of these antibiotic resistance traits. But the only thing that you and I can do is consciously reduce our consumption of antibiotic-laden animal proteins so that we force the farmers to get antibiotics out of our food. And I really applaud the fast food vendors who are really going to an antibiotic-free activity. So that's the snippet.
What? You're never going to stop them from feeding antibiotics, right?
No. It's too lucrative.
Especially outside of the U.S. China will never stop doing it, right?
That is correct. And remember, we did a paper on TWiM. That's where colistin resistance came out of, is China.
So, Michael, basically, farmers are more interested in profit than in human health. Is that, do I have it right?
They don't know. I wouldn't go that far to indict the farmer, but
they don't know.
I think this whole story of the flies really opens your eyes to what's actually going on. The other thing is we know that poultry manufacturers are really controlling who goes into the chicken farm, who goes into the turkey farm because of the avian influenza. And, you just have to buy the dozen eggs to know that we've been culling chickens for quite some time because of avian influenza. And they're really becoming anal retentive at who goes into the barns. But the flies are still going in and they're getting out.
Right. So can the flies, though, carry? So some of this avian influenza, the problem has been the wild birds. Are the reservoir?
The wild birds.
Right. So the question I guess I have is I'm sure there are papers that said this before, but basically this is really showing this connection between the antibiotic use and then the resistance at least coming potentially into your home.
Into the stool and into your home.
Right. And so that's actually a really important connection because, before it's like, well, we find runoff everywhere with antibiotics. But the question is flies, we see them in our homes. They are coming from somewhere, right? And so.
And the thing that intrigued me with the flies and the avian influenza, because in chickens and birds, it's principally a diarrheal disease. And I don't, in humans, it's a respiratory disease, but in those animals, it's a diarrheal disease. And so if the chicken gets diarrhea and the fly ingests some of the stool, is the fly actually able to transmit the virus to us? And I'm not the virologist here, Vincent is. So what do you think?
Well, most vectored viruses require replication in the vector. There are very few examples of mechanical transmission, right? Which is basically what this. Are these bacteria reproducing in the flies, do you think, Michael? They didn't look at that.
They're in the gut. They don't look at it, but
they're in the gut. So they may
be reproducing.
I think it's unlikely that a virus would be effectively transmitted because it's not going to reproduce in the fly, especially influenza viruses. But
thank goodness.
Yes.
Thank goodness. There is hope.
I mean, what I understand, I have a friend who runs a cattle ranch in Kansas, but that it's just even with they're not the feedlot, they just raise them.
Yeah.
So, I mean, and cows are a little different than pigs. They're not quite as close together even. But the antibiotics, basically, the way cattle are in the winter, they just let them kind of range and they can't get out there to treat them and stuff. So some of this is just kind of what you have to do. I think it's less of a problem because it's, cattle and lots of land. But I think some of this, the margins are really low. And if this is the only way to make money and we need our food supply. I do know Denmark actually banned the use of antibiotics in pig farming, and they actually have a higher per capita pig ratio than we do. So I believe that it can be done. It just requires changing how you do things, and that's expensive. And also, organizational change is difficult. So,
Oh, yes.
Well, Pat Brown wants to replace all meat. So we'll see how that goes.
His meat costs like $12 a pound. He's going to have to come down in price.
Yeah,
and it's delicious, but
I love it. Yeah, I don't eat meat anymore, mainly because of things like this and the way animals are treated and a little bit of health concern. But most of the world will continue, and in many parts, it's a huge protein source, right? So
absolutely, and the developing and emerging markets are actually demanding the same level of animal protein in their diet. That's all part of, air quote, growing up as a nation. And so we really need to be dropping the price of these plant-based proteins and really changing the mindset.
Yeah.
Now we have a beef substitute. I've seen chicken substitutes in the market. There's quite a few out there.
Yeah. Pat Brown wants to replace everything, all meats. But Petra, eventually the price will go down, right?
Yeah, no. I think as more competition comes up. But yeah, their chicken substitute is really good. Their meat substitute's delicious. My son is vegetarian, but was a big hamburger fan. And so we use it a lot.
I like that. It's really good, yeah. All right, Michael, thank you for the flies and pigs. Now let's hear about squid from Petra.
Something completely different.
I know. This is the most non-kosher episode I've ever been associated with.
Yes, this is non-kosher for sure.
So this paper, first of all, what caught my eye about this paper is the title. It's from so it's at Piper and MBio. It's open access. The title is Failure to Launch, Development of a Reproductive Organ linked to symbiotic bacteria. So this is essentially a story of how microbiome is important, not only in general when you're fully grown, but also for development. This is from Sarah McAnulty, Allison Kerwin, Eric Koch, Barrett Nuttall, Andrea Soria, Andrew Collins, Tyler Schleicher, Bethany Rader, and Spencer Nyholm at the University of Connecticut in Storrs. And as a little side thing, I grew up in New England, and UConn has the best, most funny mascot because they're UConn, University of Connecticut. Their mascot is a husky, even though they are nowhere near Alaska. So I always appreciate that, especially as we come into the tournament. They tend to have a very good women's basketball team. In any case, so this paper it's looking at how a microbiome in the bobtail squid, the Hawaiian bobtail squid, when everybody should look up little videos of this guy. He's the cutest, or she's the cutest. It's the cutest little animal. It only comes out at night. And during the day, it buries itself in the sand. It literally uses these tiny little arms to cover itself and hide from any predators. And then it comes out at night, and it's just the most adorable little thing. But it's most famous, actually, for being studied as a model for mutualism. So this bacteria called Vibrio fischeri, which is a gram-negative gamma protease bacteria, when it comes to high density, it actually turns on genes that make it glow. So it's bioluminescent. And the squid have developed this special organ, which is called the light organ, that is colonized specifically by Vibrio fischeri. And when they get to high density, they glow. And so the light organ of the squid glows. And as best as anyone can figure out, this is so that the squid during the night, it doesn't create a shadow against the moon for its predators below it. It's very terrified of everything, I think. And the light basically makes the predator see light above it and think, oh, it's the moon, not a squid. I don't know anything what's going through the predator's head, but that's clearly the idea of what's going on here. And this has been really famous, the Fisher. I just want to give it the credit it's due, because the colonization and the mutualism and the interaction between this squid and this bacteria has been studied. How do the bacteria recognize light organ? How do they get to the light organ? How do they colonize it? Basically in pure culture. And there's all this cell-cell signaling, both from the host to the bacteria and back and forth. And then Bonnie Bassler. So a lot of the mutualism has been studied by Margaret McFall Nagai and Ned Ruby. Bonnie Bassler at Princeton has studied the signaling. And so this idea that the bacteria, how do they know to turn on the light organ when they're at high density? How do they know when they're at high density? They actually don't. It's not like they can feel things touching them in this case. Some bacteria do, but these guys don't. They are always secreting a small molecule. And when there are a lot of them, the concentration of that small molecule is really high. And it sends a signal to turn on the genes for light expression, for expression of the LEX genes, which give them bioluminescence. And so there's all this beautiful work on the light organ and Vibrio fischeri. Okay, this paper has nothing to do with that. It will do a little control in here, but it's actually looking at development of another organ, which is called the accessory nidamental gland, or ANG. And this gland, when eggs develop in fish or chickens, you have an egg, it develops, and then it has to get out. And in chickens, it gets a hard shell put on it. But in these squid, it gets has a membrane, and then it gets this coating of a jelly-like substance that protects it. And that comes through this special gland, the ANG, and it coats the eggs. And inside the ANG are microbes, bacteria mostly, and those are in the jelly that coats the eggs. And it hasn't been studied that well as far as I can tell, but the bacteria they think are there to sort of provide a competitive environment to prevent the eggs from becoming contaminated, because essentially they're not like chicken eggs. They're not sealed off in a hard shell. They're just in this jelly, and the jelly probably tastes good to something. And so to prevent the other bacteria from growing there, they are infused with these bacteria. So it's a really beautiful organ, and it's highly invaginated. It's a really beautiful structure. If you look at figure 1 in the paper, there's a really beautiful picture of it. And also when it's not there, there's also a figure 1A is the obligatory picture of the bobtail squid. Oh, so cute. And this poor guy doesn't have any sand to hide in, so I'm sure it's not that happy. But they're very small. You can see the 10 millimeter marker in 1A. But if you look in figure B, they start to kind of show you this organ. And it's really visible, I think, in figure D. If you kind of zoom in, or figure E, you can see this highly invaginated structure. And for F, it's very invaginated and full of these tubules, and that's where the eggs kind of come through and they get this coating. There's a nice picture of it also, a kind of cross-section in G with some fluorescence, which is a confocal, so it's showing just a section of it. And you can see all these tubules, and that's where this jelly is secreted and these bacteria come in. Okay, so what they noticed, so mostly it's a lot of times people get these squid directly from Hawaii. They want to raise them in their labs, but often they just get them from Hawaii. I think they're pretty common. And when they come, they're adults, and they will lay eggs and they will develop. But as people have been working more and more on how they're colonized, they've wanted to grow them in the lab, because that way you could raise the equivalent of gnotobiotic ones that aren't infected with Vibrio fischeri or other things. And this lab, I think in collaboration with the Mandel lab at University of Wisconsin, noticed that the ones that were developing in lab, I mean, they're fine. They look like squid. Probably their light organs are fine, but they don't really produce this special organ, which is needed to make eggs. So they're essentially sterile. And when they did a sort of comparison, they looked at when they grew bobtail squid on different media from University of Connecticut or Wisconsin, they saw that they got sort of not great development of this ANG, and that they weren't probably fertile. They got kind of erratic. Like sometimes they'd kind of have one, but it wasn't great. Sometimes they'd have none. And the sand that these guys were raised on had been in the lab for over a year. It might have originally come on the original shipment from Hawaii, where these are native, but it wasn't fresh. And sometimes the labs would, add new squid and dump in sand. They weren't really that particular about it. They were just, keeping the squid alive, had some sand, dump it in, put new squid in the tank. But there was nothing really regulated. And when they looked at the bacterial community profiles, and this is in figure, actually it's not really in figure 2, but the profiles, if you look at figure...
is it 3? They have wild type, lab sand.
Yeah, that was pretty good. I actually think the best one is the Bray-Curtis diversity one, which is actually figure 5, but the diversity is really different. So in the wild ones, they usually have kind of a consortium of bacteria, so a whole bunch of different species, but 2 major taxa, the Rhodobacteriacealis, which are alpha proteobacteria, and the Viracumicrobia, which are actually relatives of chlamydia. And they noticed that when they look at the communities of these guys, they got very different communities depending on where they were raised. And so in some cases, they got much more of the Rhodobacteriacealis, and usually the Viracumicrobia were really low. But if they used wet sand from Hawaii and raised their squid, so fresh, that they just didn't let dry out, nothing super special, just making sure it stayed wet, they got what they would expect, like that were in the wild raised squid. And if they autoclaved the sand, the fresh Hawaiian, the native sand, then they didn't get egg development again. So that says something about the microbiome is really important. And this is actually in figure 2. It's actually I love this figure. I love these kinds of figures. It's very clear. They have like little symbols for heat or no heat and what they get. So in the top, they have Connecticut sand that they autoclave and then or don't autoclave and whether they have egg present. So if they've wet collected sand and they don't autoclave it, 8 out of 8. So at the top of so they sort of have the symbols at the top and then it's a table, but it's also a pictogram. So on the next one, so they get 8 out of 8 on figure 2. So fresh sand from Hawaii, everybody's happy. If they autoclave the sand, 0 out of 9 squid have an egg. It's like not developed at all. If they use wet sand, dry collected sand, so if you just skip down one, and they do that same experiment, so they're collecting sand from above the tide line, it's been sitting in the sun, it's not seen water forever, that they don't get any development of bacteria. So I think this really says that there's something special about where they're getting it from, that the wet sand from Hawaii is important. And if you have old lab sand, it's just not going to give you good development. And it's something about the community because the community changes. So one of the things that's actually in here, which I thought was a really neat experiment, is on this figure 2, they have these little blue bacteria that they add to the wet collected sand. So one of the things they're wondering is, okay, if we take the wet collected sand, the Hawaiian sand, and we have Hawaiian squid, and we let them develop, and they're all happy, and they have this microbiome, and they get the egg development, can we spike it? Can we add bacteria that should be there, but from the lab? So they actually isolate these bacteria, Lysingera, which are normally in the wild-caught squid. And they pick them, and this was very smart, because they form these really blue colonies, which are really easy to isolate. And so they grow in the lab, and they're blue, so you can get pure culture, and you can spike the wet collected sand, and you can kind of track them. And they see that if you use fresh sand, you can add you can add these guys to the microbiome, and they get incorporated really nicely. They get 7 out of 7. So this is the 3rd arrow down on this little chart. If they do that with dry collected sand, they don't get any colonization. If they kind of use a mixture, they can't get it to colonize. So there's something special about the big consortium that they get out of the native environment where these guys are normally found. So they're missing something. They can't just add back what they pulled out. It's something probably small, but very, very important to get egg development. One of the questions that they had, which is really important because, again, it might not be important except for bobtail squid have been the place to study Vibrio fischeri. They do the experiment to see maybe this thing might be Vibrio fischeri. Maybe Vibrio fischeri are important. So they actually essentially see if Vibrio fischeri are important, and it turns out actually they're not. It has nothing to do with the light organ. It's really only this microbiome and this development of this special reproductive organ, which I think is really neat. So now the squid is like, oh, we can use it for one thing, and we can also use it for consortial microbiome mutualist interaction, which is really great because we don't know that much about consortial microbiomes in terms of development. The human microbiome we've talked about before is very complicated and difficult, but here it might be more simple, but plus you can also use this little squid as your model. I think that was a really important control and really easy for them to do. Okay, so one of the things is they're like, well, what's happening? We get fresh sand, it's great. We get sand from Wisconsin or sand from Connecticut, that we basically, the sand is just old sand, and we don't get what we want, or we don't get reproducibility. And so they look at taxonomic diversity. And I think this is really interesting. And I think this is important in general that people think about this when they do microbiome studies. This is, I think, one of my favorite figures for diversity, mostly because sometimes these figures are very complicated, and this one is really simple. So this is figure 5. It's a Bray-Curtis beta diversity analysis, so pretty standard kind of analysis. And they're looking at the diversity in, so they have these two, the X and Y are sort of where the diversity is, what kind of diversity it is. So all you need to know is that certain groups mean they're basically the same kind of diversity, the same things are in it. It's about the same proportions. So if you look at the wild in sort of this royal blue, it's in the lower, bad with left and right, lower right quadrant. And that's great. But if you look at the Connecticut lab sand or the Wisconsin lab sand, not only are they, the Wisconsin lab sand is on the bottom, but over to the left, and the Connecticut is near the top. And over to the right. So not only are the Connecticut and Wisconsin not at all the same diversity as the wild, they're also in two different places. So they've diverged also from each other. So even just having old sand, it doesn't always end up in the same place. And I think that's also just something to think about when you have bacteria and you have a culture, right? It might not be the same. This happens all the time with stocks, like the bacteria that we have stocked, our wild E. coli MG1655, we got from WashU Med, but is that like the one my colleagues at Wisconsin use because of mutations and things like that? And here you're looking at a whole consortium of bacteria, just a few things like if one starts to dominate or, who knows, you can get divergence of these communities really fast. And then what's really nice about this figure is they have in pink, they have the squid that were born in Connecticut but raised on sand from Hawaii, fresh sand from Hawaii. And their diversity, you can already see it's diverging. It's kind of moving up the y-axis, but it's still much closer to the wild caught squid than it is to the other, the old Connecticut or old Wisconsin, old Wisconsin sand. And again, it's only a year old, but that's a long time for bacteria. And I thought that was just really beautiful because it's really showing like how fast these things diverge. And also, I think probably if you're trying to raise squid, you want fresh Hawaiian sand. Maybe you want to work at University of Hawaii, Manoa. But in any case, it definitely can be done in Connecticut. You just need to get the good stuff. So I thought that was really interesting. And then in figure 6, they actually look at the diversity a little bit here. And this is to just look at laboratory months. So they take the fresh sand and basically over 6 months, they look and see how the diversity changes. And what they see is that the Rhodobacteriaceae taxa, so they're using 16S. So this is fairly low resolution, but it's actually working really well for them. They see it increase with time, and so do the flavobacteria. But these Viracomicrobia, they decrease to almost undetectable levels in their experiments. So the question is like, which ones are important? They don't really know, but it's clear you're getting major changes. And, they have some variation in their results and like how much things change, but it's clear like these are probably dominant patterns. The flavos and the Rhodobacteriaceae increase and these others decrease. And so the overall message is the specific taxa are really important, but there's probably a few things that might be members of taxa, because again, this is a very high level analysis with 16S, but it might also be, there might be small things in there that are really important. They don't know what it is that's actually doing it, but it's like you can have a consortium, you can look at it in development. And this is really exciting because essentially now the squid can be another model for understanding development, and it's a pretty easy readout for them.
Light or no light?
Well, light or no light, exactly. You don't even have to deal with the Vibrio fischeri. I'm not sure it's good for the squid, but again, I don't think these are very endangered or anything. And they have a model. Figure 7 is pretty cute. They show the egg development, but basically it's showing these environmental bacteria are really important for the development of this little tubulated organ that puts on this gelatinous stuff. But they make the point, like, we can now start to pick apart. Maybe we can find what to add. I think it's going to help people who are trying to raise these squid in the lab, because going to Hawaii is expensive, getting lots of sand. I mean, this is a decent sized tank. And a lot of these labs have 20, 30 tanks of these things. You need enough sand so the little guys can bury themselves. That's what their behavior. And I think it'll be really helpful. I think it's also really important because you can also study on the squid side. So this is sort of Margaret McFall and the guys kind of side, that you can start to study what are the signals that are going on between this organ, the egg, and the bacteria. And if you can identify which bacteria is the magic one, you could really start to pick it apart and make mutations in the bacteria, kind of like they've done. It's more complicated than the light organ, which is basically pure culture, but they can go back and forth. And again, this is really important because a lot of things about our microbiome, we know that gnotobiotic mice that haven't been exposed to any microbes, so they don't have any of them on their gut or their skin, they don't develop that well. Being exposed to microbes, we were talking about flies and being dirty, right? You need to be exposed to microbes actually in order to develop a good, healthy immune system, because your body needs to recognize different things when it's really in the immune development stage. So you don't want your baby's environment to be completely pristine. This is a way of kind of looking at that, because it's not about the immune system. But if you think of the immune system kind of like a complicated giant organ that's not solid, you can think about this in the same way. It's a pretty complicated organ. It's got lots of signals going on, and these bacteria are helping it to develop properly. And so I think that's really exciting in the system. As far as I know, the light organ develops fine without the Vibrio fischeri, but here you really need this for development. And so that's you have an established model organism, the bobtail squid, and now you can look at something else in it. And I just loved it. So, and again, the title, you can't beat failure to launch as your title.
No.
It's great. Well, we heard from Sarah, the first author of this paper, Sarah McAnulty. A couple of things about Sarah. Her path to science, she was interested in animals from being a kid, but didn't know what scientists did. Her parents also didn't know. So they thought she should work in a zoo or an aquarium. They were super supportive, but none of us really had a connection to science when I was growing up, she writes. I had no idea squid biologist was even a job until my sophomore year of college, when Lydia Matker from the Marine Biological Laboratory gave a talk at the Marine Science Department of BU, Boston University, where she was studying. The following summer, she worked with her in Roger Hanlon's lab. After graduating with a marine sciences bachelor's degree, Sarah realized she really wanted to be working with squid immune systems, but her undergrad degree didn't have a lot of molecular and cell work. So she worked 2 years in Germany, filling the gaps, started in Spencer Nyholm's lab in 2013. And she writes that they started raising the squid for this project in 2014. I remember Margaret talking about how long it takes to raise sufficient squid for these experiments. As for critical contributions by other people in her group, she provides a Twitter thread, which includes an overview of the other major contributions, namely Allison's sequencing work, which was huge for this paper, Andrea Surya's isolation of bacterial strains that are used in inoculation experiments, including critically the experiments where they were able to incorporate the blue strain, JC1, into the ANG when raising squid on sand from their Hawaiian ecosystem. So we'll have a link to that. As for strong memory, figuring out the right conditions to raise these bobtails that would induce ANG development took years of work. Every time they raised a cohort, the baby squid took over her life. Not that she's complaining. She loved raising squid, but it was an all-consuming effort. She'd be down in the squid room hours every day, caring for these tiny little squid as they turned from apple seeds to blueberries to grapes. On the day they finally got an A&G, she had raised 4 or so cohorts of squid that didn't develop A&Gs. They were pretty used to failing at this point. Getting squid to sexual maturity took 2 months, so each reveal day was weeks in the making. She brought the squid up to the lab, held her breath as she looked into the first squid. Her lab mates were all there, and Spencer, the PI, was there for the grand reveal too. When they saw the first A&G, they lost it. They were stomping around the lab. Everybody was high-fiving. One of the best moments she's ever had as a scientist. And Sarah wants you to know about Skype a Scientist, an informal science education nonprofit that focuses on bringing science to as many people as possible through personal connections with scientists, public art. They match scientists with classrooms, libraries, scout troops, and other groups for Q&As to talk about science. They want to make science accessible through personal connections, conversations, not just lectures. Sarah thinks the most effective science communication is done through two-way conversation, not just talking at people. That's the core of what Skype a Scientist does. It's something I thought of 15 years ago. I thought conversations are the way to learn. And
hence the podcast.
Yeah, but I had so much trouble getting people at big meetings to get that.
Yeah.
You know?
I think this is actually a wonderful thing, Skype a Scientist. And I encourage everybody to look at Sarah's Twitter feed, which maybe we can put on there because she advertises it. But if you're interested in participating, they're always looking for volunteers. But really, it's like directly connecting scientists with, a troop of scouts. That sounds awesome.
That's good. In addition to science, which she writes is admittedly on the side to my main career as a science communicator, she is executive director of Skype a Scientist. She makes squid-themed art, plays water polo, and is involved in her community in Philadelphia, and is vice president of the Fishtown Neighbors Association, which does community work, including mutual aid, managing public art projects, and connecting neighbors for a stronger community. And finally, how about advice? She generally tells folks to follow all of their interests, especially those that aren't directly at the lab bench. We need scientists to be mentally healthy, and focusing exclusively on one topic all the time can, for some folks, be damaging over time. Science is stronger when we let our neurons have a day off and make connections that we may not have the opportunity to make when only interacting with folks who think like us and work on the same topics as us all the time. Just this morning, I was in a workshop with grad students talking about communicating science via art. They each made a drawing of their experiments, and one of the scientists said, "Wow, I look at my plants every day, and I didn't even know the leaf arrangement until I was drawing them. What are we not noticing about our study organisms by studying them the same way all the time? A variety of experimental approaches is always important, but additionally, these abstract artistic approaches can be just as powerful for helping our brains make the connections that lead to major breakthroughs." It's good advice. Take a break. Do something else every now and then. Mm-hmm. All right. Thank you, Petra. Very fun paper. That'll do it for TWiM 284. Michael and Petra, you think we'll ever reach 1,000? Probably not.
No.
We do one a month. We do 2 a month. That's
we do about 26 a year.
26 a year. Nah. No. 10.
Maybe somebody will. But I don't know if it'll be
26 a year. No, I'm not going to be alive for 1,000. Isn't that sad, isn't it?
Well, we could have a bacterial outbreak like COVID, and then we'd be doing 3 a week like you did.
1,000. I'm going to do the math. 1,000. We're at 284. So that leaves us 716 divided by 26 a year is 27 and a half years.
Yeah, no.
Well, Petra, you'll be around. I'm not.
I hope so, but
maybe you're not going to be doing TWiM.
I don't know.
But I hope other people will take over. That's my goal for all of these pods, is to have people take over. And we'll see how that goes. Anyway, TWiM 284, the show notes are at microbe.tv/TWiM. You can send questions and comments to twim@microbe.tv. And if you enjoy our work, we would love to have your financial support. Microbe TV is a nonprofit 501(c), so your contributions are tax deductible. You can go to microbe.tv/contribute. We have a number of ways that you can help us out. Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And Petra Levins at Washington University in St. Louis. Thanks, Petra.
Thank you.
I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.